In many respects, the central function of life is to produce more life. And the central problem in reproduction is information. How do you pass information from one generation to the next? One of the great discoveries of genetics is that each parent contributes genetic information to offspring. In the first half of this lecture, I'm going to set the historical stage of genetic research by introducing the concept of genetic information, by describing selective breeding of livestock and plants, and examining spontaneous generation. Then I'm going to tell you about the seminal work of the Czechoslovakian monk Gregor Mendel and his 4 laws of classical genetics. These 4 laws are: first, there exist genes which carry traits. Second, that each individual carries 2 genes associated with each trait, 1 from each parent. Third is that genes come in different forms, or alleles. There are dominant alleles, which are expressed in preference to recessive alleles. And finally, the different alleles are sorted and distributed randomly. All combination of alleles, therefore, are equally likely in Mendel's genetics. So let's begin by setting the historical stage. In the last lecture, we saw that cells may be thought of as the fundamental building blocks of life. All life is formed from cells, and each cell acts like a chemical factory, taking in raw materials and energy, manufacturing various chemicals, including all the chemicals that are necessary to make copies of the cells themselves. But here we run into a real mystery. All cells come from prior cells. That's a central tenet of the cell theory. But how can a tiny cell carry all that information that's needed to make copies of itself? And how can cells then pass that information on to next generation of cells, on and on and on, in an unbroken chain? In a sense, therefore, the central problem of life is a problem of biological information. Without information, there can't be life. To construct a living thing requires vast amounts of information about molecular building blocks, about body architecture, about the dynamic processes by which organisms develop, by which they reproduce. In a sense, all life forms represent solutions to this problem of passing on genetic information from one generation to the next to the next. Genetics, therefore, is the study of the ways by which this biological information is passed down from parents to offspring. Think about the reproduction of a human being. At the instant of conception, all of the information necessary to create a unique human being is contained in that fertilized egg. That's an object that's just 1/100th of an inch across, and yet it contains every bit of information that makes you and me and everyone else a unique human being. As that first cell divides, an embryo starts to develop. The coded information, therefore, has to be duplicated and read time and time again, and it has to be used to manufacture all the different components, the fingers, the heart, the brain of a human being. Each individual may eventually pass a portion of his or her unique genetic message then to subsequent generations, and the same genetic code then is passed down from one generation to another in the same way. This process of information transfer has continued unbroken from the very first living cell nearly 4 billion years ago, and it's led to the entire diversity of modern life. How can this be? Where did that information come from? For centuries, scientists and philosophers have wondered how the vast amount of information necessary to produce a living organism could be stored, could be duplicated, could be interpreted in an object as tiny as a cell. Every time a cell divides, that information gets passed on, and that's the central mystery of genetics. By early in this century, biologists agreed that the genetic message is somehow carried in structures called chromosomes. These are elongated cellular structures that duplicate and divide, and they do so evenly just prior to every cell division. Researchers also in this period concluded that the information must be coded into some kind of molecular structure, a molecule being the only thing small enough to hold information and still be inside a cell. The smallest cells, remember, are only a few thousand molecules across. So a molecular structure has to be the thing that holds that information. And we don't know of any other kind of information storage mechanism. This was, again, the great mystery in biology. As our understanding of life's genetic mechanisms deepens, we're beginning to learn ways to modify life, to actually engineer that genetic code at the molecular level. This new knowledge is powerful. New genetic technologies hold fantastic opportunities and also tremendous risks. But before embarking on this investigation of genetics, we really have to set the historical stage. We have to look at what came before the modern age of molecular genetics. The history of genetics can be divided into three major eras. First came classical genetics. That's studying of genetics at the level of organisms and their traits. Then came cellular genetics, when microscopes became good enough that we could actually look inside the cell and see what was happening as cells divided. And then finally, molecular genetics, looking at genetics at the scale of the molecule that contains the genetic code, DNA. We're going to begin with classical genetics, which looks how biological information is passed from parents to offspring at the level of complete organisms, animals and plants, for example. Humans have studied this for thousands of years because humans have taken advantage of a simple fact of life, that like begets like. In humans, just as in other animals and in plants, offspring bear a resemblance to their parents. If you've seen a family photo album, you can tell immediately people are related. Parents and children, cousins, aunts and uncles, and so forth. Families have relations because of the passing down of similar traits. And this phenomenon has been exploited since prehistoric times in the selective breeding of animals, domesticated animals such as sheep and dogs, of course, horses, all the different varieties of these animals that we use in various ways. Sometimes breeders took advantage of sports of nature, they were called. These are mutants that had particularly unusual qualities, dwarf animals, for example, or giants, particularly fertile animals or particularly productive ones in one sort or another. People have used these in selective breeding to develop new types, new varieties of organisms. Many new varieties of cultivated plants were developed all around the world. Rice, wheat, and barley were improved in Asia. We had yams, peanuts, and gourds that were selectively cultivated in Africa. Corn, squash, and peppers, tomatoes in the Americas. Interestingly, there's no record in prehistoric times of anyone in Europe doing selective cultivation. So it was not universal, but very widely adopted around the world. Well, not surprisingly, this idea of inherited traits was applied to human beings. Ancient scholars noted that certain diseases run in families. Hemophilia, for example, arthritis, certain kinds of mental disorders ran in families. Some societies also concluded that undesirable characteristics such as dishonesty or sloth, cowardice, also ran in families. And this observation may have led in some societies to the caste system, where highborn and lowborn actually represented a prediction of certain personality traits, which we certainly wouldn't agree to today. The context of ancient thinking about genetics was quite different from today. The doctrine of spontaneous generation is a tenet of the teachings of Aristotle. It claimed that inanimate matter was imbued with a life force, and so that life spontaneously arises around us all the time. This idea hindered genetic understanding because if maggots appear spontaneously and other life forms can just occur from inanimate life, then somehow that information was contained in a kind of ether, in a way that you can't say was physically passed from one cell to another. The relationship between flies and maggots, and indeed the dismantling of the spontaneous generation theory, therefore was critical to genetic understanding. An important step was taken by the Italian naturalist Francesco Redi, who lived from 1626 to 1697. In the year 1668, Redi published the results of a whole series of experiments that showed that meat, which was kept isolated, which was kept away from insects, never produced other insects. That is, maggots did not form in this way. So this helped discard one of the tenets of spontaneous generation about maggots. And then there were many other important contributions as well. Some were made by the amateur Dutch scientist Antony van Leeuwenhoek, who lived from 1632 to 1723. He was a contemporary of Redi, and of Robert Hooke. He also used microscopes and was the first person to look through the microscope to try to understand the duplication of life or the origin of life. Leeuwenhoek was a fascinating person. He was familiar with magnifying lenses that were used by drapers to examine cloth. That was in his native Holland. And he improved on their magnifying lenses with ground glass lenses of his own construction, exquisitely built lenses, much finer than it is said than anyone else at the time. And he made hundreds of microscopes in his lifetime. The best of these microscopes weren't even equaled until the 19th century. For 50 years, he applied these technical skills in designing the microscopes, and then his unusually keen eyesight was used to explore the microscopic world. Eventually, because he didn't have a formal academic training and any degree, he didn't engage in the kind of speculation that characterized a lot of the work of that period. So what he did is he simply made observations, very meticulous observations, and he reported what he saw, often to the Royal Society, often to Robert Hooke, for example. Van Leeuwenhoek was the first to observe microorganisms, single-celled organisms in water. He observed the sperm cells in semen also, the first to do that. And he suggested that every sperm contains a complete miniature human being, so that when the egg is fertilized, that human being is inserted into the egg and starts to grow in that way. Much of Van Leeuwenhoek's efforts were directed towards a refutation then of this doctrine of spontaneous generation. And it was his work, along with that of Redi and many other people, including Louis Pasteur, that finally debunked that whole idea. So then you had to contend with the fact that biological information was passed from one generation of cell to the next, a key tenet of the cell theory. Now, in the 19th century, the role of parents in genetics was becoming clear, but there was a major mystery. That is, how does a fully formed plant or animal develop from that single fertilized cell? You needed to have a new experimental approach. And that approach was provided by the Czechoslovakian monk Gregor Mendel, the founder of modern genetics. Mendel, who lived from 1822 to 1884, grew up in a home with a father who was a farmer. The farmer had served in the army during the Napoleonic War. His mother and many close relatives turned out to be gardeners, and this certainly influenced his future career in science. As a dedicated student, Mendel suffered financial setbacks that interrupted his studies time and time again. So he finally entered the Augustinian monastery at Brno, which was then in Austria, but it's now part of Czechoslovakia. He did this at the age of 21, and he took the name Gregor. Though he didn't really feel a calling to holy orders at the time, Mendel found that there was peace and security, an environment that he needed to continue his studies. And that's why he went into the monastery, at least initially. This particular monastery actively promoted study and learning, and it had an experimental research garden of all things. Several monks taught at the local philosophical institute, and during his training at the monastery, Mendel learned much about the experimental method, the scientific method which he could apply to the study of genetics. He engaged in experiments on sheep breeding, on hybridization of plants. He also was allowed to study natural history. He was sent for a time to the University of Vienna, which is quite remarkable if you think about it for a Czechoslovakian monk. Late in his career, Mendel was elected as abbot of the monastery, and for the last 16 years of his life, he was consumed by official duties, especially conflicts with the local political bureaucracy. So if you think about it, his career as a scientist itself was very short. Indeed, his most famous experiments were done over a period of only about 8 years in his entire life. Mendel's famous genetic research on pea plants was conducted between 1856 and 1863 in that monastery garden. What he did was this. He studied pea plants, and he focused his attention on 7 distinctive traits of pea plants. In particular, he saw that some pea plants were short and some plants were tall, never in between. Some pea plants had white flowers and others had purple flowers. Some had smooth pea pods, some had wrinkled pea pods. Peas always displayed one trait or the other, and there were 7 of these traits in total. This is unlike quite a number of organisms in which they're intermediate traits. For example, humans don't come in just 2 varieties, short and tall. There's all different sizes in between. But these pea plants that Mendel was studying were either short or tall. a great advantage when you're trying to study the origin of traits and the passing of traits from one generation to the next. Mendel started by establishing populations of purebred plants. These are populations that always bred true. Short plants that when crossbred with each other always produce short plants. Tall plants which when crossed with each other always produce tall plants. And he kept these populations separate to begin his experiments. And so he had a whole series of populations that showed these different purebred traits. Pea plants are normally self-pollinating. That is, the plant actually produces pollen and accepts the pollen from itself, and new pea plants are then sort of self-produced, a kind of cloning mechanism, if you will. But Mendel removed the male sex organs from the plants that he wanted to study, and he hand-pollinated these plants with pollen from a pre-selected father. So the male pollen was then carefully transferred from one plant to another. He began his experiments by studying a single trait, for example, tallness. He'd take a whole group of plants where he'd crossbreed a purebred tall plant with a purebred short plant. And in the first generation of these experiments, every one of the hybrid plants of that first generation turned out to be tall. Mendel then called this trait, the one that appeared in the first generation, to be the dominant trait. He said that dominant was, the tall plants were dominant, short plants had the recessive trait. I should make a point right here that recessive and dominant traits do not mean better and worse. And that sometimes can be confusing because dominant sounds like stronger. There are many dominant traits that are quite destructive. In genetic diseases, if you have a dominant trait that's a genetic disease, it can kill you. So the idea of dominant and recessive only has to do with whether or not a trait is expressed, whether it appears in the organism. Okay, so that's the first generation, the hybrid generation of a tall and a cross short, everything's tall. But here comes the interesting part. When you crossbreed those hybrids with each other, you then find that 1/4 of the offspring on average are short and 3/4 are tall. In the next generation, the short plants that are crossbred with each other only breed short plants. So this is in the third generation now, but the tall plants when crossbred give you a combination of short and tall. So you get a rather complex sequence of events. Mendel then did a whole series of experiments where he considered combinations of traits, two traits at once. For example, round versus wrinkled seeds and yellow versus green seed color. What he found was that the round shape of a seed and the yellow color are the dominant traits, so wrinkled and green peas have the recessive traits, and he did crossbreeding of those. What he'd find is that when he had purebred round wrinkled plants and purebred yellow-green plants, when you crossbred them, the first generation of hybrids were always round yellow peas. But then in the next generation, you found a distribution that out of every 16 plants, on average 9 had round yellow seeds, 3 had round green seeds, 3 had wrinkled yellow seeds, and 1 out of 16 had the double recessive trait of wrinkled green seeds. Mendel showed that this distribution is exactly what you'd expect if the two traits were completely independent of each other. Believe it or not, Mendel performed more than 28,000 individual experiments to determine this kind of behavior in plants. From this vast accumulation of data, he derived 4 key ideas which might be called the 4 laws of classical genetics. First law. This states that there exist what Mendel called atoms of inheritance, what we call genes now, and these genes carry traits. In peas, there's an atom of inheritance for tallness. There's an atom of inheritance for shortness, and so forth. Mendel didn't know what these atoms were. He knew that they must be physical structures of some kind, but he didn't have any idea what they were. He knew that they had to be very small because they fit into cells, but other than that, who knew? Second law is that each parent contributes half of an offspring's genes. Each individual therefore carries 2 genes for each trait, one from the father and one from the mother. And this was a dramatic demonstration of a concept which had been much debated. Whether the mother contributed genetic information more than the father or vice versa, that had been a matter of philosophical debate for a long time. Mendel said it's equal, half from each. The third law is that genes come in different forms, what we now call different alleles. Some alleles are dominant some are recessive. Tall is dominant, short is recessive. When both alleles are present, the dominant one is the one that's always expressed. And Mendel's fourth law, which actually holds true only in special cases, he was lucky that pea plants happen to do this, is that these different alleles are expressed independently, randomly of each other. So all combinations of alleles are equally likely. These principles of classical genetics can be illustrated in a square diagram. And at this point, those of you who are listening on audio tape might want to take a look at your booklets. If you're driving, wait until you get home or till you're at a stoplight to look. But these illustrate some of these square diagrams, and it's easier to follow if you can actually see one of these diagrams while you're listening to this. Two genes of one parent are listed on one side of the square, the top side, for example, and two genes of the other parent are listed on an adjacent side, for example, on the vertical side of the square. If you crossbreed a purebred tall plant, it's designated with two capital T's on the top, while a purebred short plant is designated with two small t's on the vertical. And then you see what the cross is, and it turns out when you have two capital T's on the top side, two small t's on the vertical side, every combination is a capital T, small t, a combination of the tall and the short allele, and therefore the tall is always expressed. Every offspring is a hybrid, and it's designated by capital T, small t, indicating the tall and the short allele together. Now, when you crossbreed two of these hybrids, on the top you have a capital T and a small t in the square. The vertical, you also have a capital T and a small t, and this leads to a different combination of letters. On the average, 1 out of every 4 offspring has 2 capital T's, 1 out of every 4 has 2 small t's, and 2 out of every 4 have a capital and a small t. That is the mixed hybrid type of arrangement. And that's why you see 1 short, 3 tall on average when you crossbreed the hybrids. A more complicated square occurs when you try to match up 2 traits. You'll see you have a 4 by 4 square in the booklet, and this leads to 16 different combinations. As we said, you have, for example, smooth and recessive smooth traits for seeds and wrinkled traits, yellow seeds, green seeds, and so forth. And you see those different combinations that are possible. But ultimately, if all combinations of 4 alleles is equally likely, the average number of plants is going to end up being 9 plants out of 16 will have smooth yellow seeds, 3 will have wrinkled yellow seeds, 3 on average will have smooth green seeds, and finally, on average, 1 out of every 16 combinations will be wrinkled green seeds with the 2 recessive traits. Mendel's results were nothing short of revolutionary. And you might think that they would have transformed biology right then and there. But it's not what happened. And there are a number of reasons for that. First of all, Mendel announced his findings at a meeting of the Natural Science Society in Czechoslovakia in March of 1865. And probably this was a meeting with lots of people discussing, and most people weren't thinking about genetics at that point, so his results really didn't resonate with anybody in the audience, at least in any major historical way. His paper was published the following year in the Society's widely distributed, but I guess seldom read journal, and that, of course, also led to its being ignored for a long time. Indeed, it was completely unappreciated in Mendel's time, and these great discoveries in genetics were not fully rediscovered until about 1900, when 3 different European scientists independently reproduced his results. And when they did a literature search, they found Mendel's paper from 35 years earlier, and must have really said, oh my gosh, you know, I should have just looked at the literature. But that's what happens. Who reads 35-year-old literature to find out current research developments? Mendel was way ahead of his time. Well, many aspects of classical genetics can be applied to our own species and the understanding of the passing of genetic information in humans. Some human traits indeed display exactly the kind of traits that Mendel discovered in pea plants. We know that brown eyes, for example, are dominant over blue eyes. But 2 brown-eyed parents that have their recessive blue-eyed genes can expect to have about 1/4 of their offspring with blue eyes. So it's certainly possible to have brown hair, brown eyes, and to come out with a blonde-haired, blue-eyed child, but only about 1/4 of the time. 2 blue-eyed parents, though, generally will not produce brown-eyed children because both parents would have the recessive blue-eyed trait, and therefore they produce blue-eyed children. There are other traits of this sort. The ability to curl the tongue is dominant over the inability to curl the tongue. Surprisingly, extra fingers, if you have a sixth finger, that's a dominant trait over 5 fingers. So if 2 people with 6 fingers were to marry, there'd be a good chance of them having children also with 6 fingers. In fact, some of those children might have 2 dominant traits, and therefore all of the offspring from then on of that individual would have the 6th finger as well. Turns out that color vision is dominant over color blindness, but there are also a number of genetic diseases that are dominant. As we shall see, for example, I'll tell you about my wife's family, which has a dominant form of a gene that leads to colon cancer. And so generation after generation, this dominant gene has been passed on and has been a killer in her family of people at fairly young ages. Having said that, there are many human traits that are not tied to one gene, and it's a fallacy to think that human behavior or human characteristics can always be identified with a single gene. That's a kind of reductionist thinking that just doesn't work. Tallness and shortness in humans, for example, is a very complex function of many, many genes. There's taller than average parents are generally going to have taller than average children, as well shorter than average parents generally have shorter children, but there are many other factors as well. And some of the traits that we are most interested in, physical attributes, intelligence, behavioral characteristics, while they may be influenced by combinations of genes, they're also influenced by environments and other developmental factors in complex ways that we really don't understand. Indeed, it turns out in retrospect that Mendel was quite fortunate because he chose pea plants. And pea plants are one of the few organisms where these characteristics turn out to be very independent and form very clear one or the other types of arrangements. Well, to summarize this lecture on classical genetics, I've tried to introduce genetics as the study of biological information, the idea that it transfers information from one generation to the next to the next. For thousands of years, humans have taken advantage of this understanding that information is passed from one generation to another in developing new breeds of animals, in developing new varieties of plants for cultivation. But until that theory of spontaneous generation was at last soundly renounced and shown to be wrong, it was very difficult for any sort of systematic genetic understanding to carry forth. It was Gregor Mendel who established then the classical laws of genetics. And it was his studies of pea plants that led to this. The study of pea plants that had very specific traits, characteristics that you could study an either-or situation, dominant or recessive, and easily verify this in a whole series of experiments. Gregor Mendel came up, therefore, with 4 laws of classical genetics. First of all, there exist traits, what he called atoms of inheritance, what we now call genes. The second law is that each individual carries 2 genes associated with every trait, one of them from the father, one of them from the mother. Equal participation in the genetics of the offspring. The third idea is the genes come in different form, the different alleles. You have dominant alleles that are expressed. You have recessive alleles that are not expressed unless both alleles are recessive. Therefore, if a dominant and a recessive allele are crossed, on average, 3/4 of the offspring will show the dominant trait, and only 1/4 will show the recessive trait. And fourth and final, the idea that different alleles are sorted and distributed randomly, that all combination of alleles are equally likely, although we now realize that in humans and in many other organisms, this is not a general law that follows. You have to be very fortunate in choosing your case and your traits for these traits to be expressed independently. What Mendel didn't know, and what scientists up until very recently did not know, was how organisms pass their genetic information from one generation to the next. What's the physical structure of genetic material? How is it physically passed from an organism? It has to be some kind of structure, some kind of chemical, some kind of molecule. In the next lecture, therefore, we're going to study the passing of genetic information from one cell to the next.